Thanks. Thanks, everyone. Can you hear me all right? Okay, cool. It's a tough act to follow, John. So my name is Michael Reiche. I'm coming from the University of Cape Town, where I work at the African Microscopy Initiative. And I thought in my talk, I want to check, I see Precious has run out. I don't know how much time we're behind schedule. So I'd encourage anyone to just stop me at any point when we need to finish this talk, because I like to talk. So pardon? 10 minutes, 20 minutes, okay. So in my talk today, I want to give a little bit of insight, not necessarily exclusively into what we're doing at the African Microscopy Initiative or what we are, but also a little bit into just some of my thoughts and opinions as to what we've seen and how this information can be helpful and fed forward to the rest of the community. To those of you who don't know, the African Microscopy Initiative is a multi-pronged effort to try and increase the utilization of advanced microscopy and research-grade microscopy in Africa and in the questions applicable to research in Africa. And we're doing this through three main kind of mechanisms. The first is an open access imaging center that we have launched at the University of Cape Town. The second is by facilitating training and education, mostly through workshops, but in any mechanism in which we can, we try and do that. And then infrastructure and development, because I think a running theme that we'll see in undertones during today's discussions is that microscopy is incredibly broad. There's a lot, and putting effort into one area is maybe going to get a little bit of an impact, but if you really want to be able to make genuine strides as a continent, we kind of have to invest in a lot of areas, both geographically, but also in terms of the pipeline, right? We can't just invest in high advanced super duper microscopes. We also need to invest in kind of stepping stones and bridges as well. So a quick overview to those, again, who aren't familiar, and I apologize if you've heard about AMI before, but for those of you who aren't aware, we have the imaging center, which is located at the University of Cape Town. And this is a BSL-2 laboratory that we try and facilitate microscopy applications to anyone on the continent that has a good justification. So it works through a project proposal and review system. So if you have an idea and you would like to use one of our microscopes for a research project, you can work with us in developing your project proposal. It will be reviewed, and then we will pay for you to come to Cape Town. We'll pay for your time in our laboratory, and we'll help you conduct your experiment. We've hosted a series of workshops. I tried to put it into numbers the other day. I think we've hosted 7 or 8 workshops in the past 3 years and trained 136 workshop participants in a range of topics, from fundamentals to sample preparation, to light microscopy and advanced techniques. We have the Microscopy Matters webinar series, which is currently just 3 online videos that deep dive into specific topics related to microscopy. We have the Partners in Teaching, which is recently announced and currently under review, and this is a mentorship train the trainer program that I'll speak a little bit more about. And then the Program for Equipment Exchange and Reutilization is a mechanism through which we're trying to get equipment to where it can be most impactful on the continent. And I see Agath is here, one of the recipients of the 4 microscopes that we helped coordinate donations for. So starting off with the AMI Imaging Center, as I mentioned, back in 2023, we had the mechanism by which we had an open call. So anyone on the continent was able to write a project proposal and submit it, and to which it was reviewed. We're currently not open for another call at the moment until we can finalize some funding details. But I thought it would be interesting to feedback some insight from that first call and what happened behind the scenes. So a contextualizer that the impetus behind the imaging center in Cape Town, the AMI Imaging Center, is that globally we have access as scientists to incredible technology that allows us to gain insight into a variety of different biology. Okay, with 3D imaging, There's structural imaging with electron microscopes. There's a plethora of really, really cutting edge things. These microscopes there might only be one of them in existence, but they can provide profound insight into various topics of cancer, infectious diseases, you name it. The problem is that within Africa, it's not the problem, it's the reality. Within Africa, we have a lot of research priorities, but currently the gap between what we fight with on a daily basis in Africa and these advanced techniques is just too massive. It's too difficult to get research projects to a point where they're using these bespoke instruments to gain this insight. So the AMI Imaging Center wants to help bridge this gap by making it a little bit easier to get to these things by doing intermediate work. The imaging center itself is one lab in which we have 5 microscopes. The entire lab is BSL2, so that allows us to work with cell culture and certain pathogens. And the intention is that everything that we need can be done in the lab. So we're doing immunofluorescence, can be done in the lab, the labeling can be done in the lab, sample prep can be done in the lab. We have the 5 instruments that span a scale or range of scales from super resolution advanced microscopes that allow us to see kind of the architecture within the biological sample. So this is at scales of 50 to 80 nanometers. We have microscopes that allow us to see biology in 3D, where we can do optical sectioning to get information, and not only the X and Y coordinate, but also the Z coordinate. So we can see the full shape and structure of biological tissues or biological cells or intracellular features as well. We have microscopes that allow us to look at live samples, so we can see how they respond and how they move and how they change to different environments. We have a microscope that allows us to high content image in a multi-well plate, so we can assay a lot of different conditions quite quickly, so in a high throughput context. And then finally, we have a slide scanning microscope that allows us to image, or I think what's traditionally referred to as whole slide imaging. So if we have histopathological samples or immunofluorescence samples, and they're coming from a mouse model or from patients, and we have 400 samples, we need to find a way to process and go through all of those samples quickly. We have a microscope that can help us do that. But it's not only about the microscope, as I briefly mentioned. There's also the lab, the sample prep, the data analysis. We also provide the infrastructure needed to do that. So we're trying to provide the whole package that can be used by any researcher on the continent. So the insights into the call that closed in 2023, and we've been working with since 2023. We had 63 applications from across the continent. You can find your country on the map and see if anyone from your country made an application. But the real insightful thing is that a majority of the proposals that were sent to us were focused on basic biology, understanding fundamentally what was happening within a sample, which is exactly what we were looking for. We're not really looking for diagnostic interventions, because that's not our strength and the capacity that we can fill. But within the basic sciences, we had a plethora. We had pathology, infectious diseases, we had cancer, we had neuroscience. There's a huge diversity, and I think that's one of the first take-home messages, is that microscopy is a bit of a double-edged sword. You can do a lot with a microscope in a lot of different fields, and it's a hugely utilized or impactful system. But somebody who's done a neuroscience project is only going to be able to convey a certain amount of information to somebody who's doing an infectious disease project. Okay, so in addition to the microscopy, there needs to be a firm understanding of the biology and the rationale behind what you're using the microscope for. Okay. Of the 63 applications that we received, a little more than a quarter of them sought to do live imaging, so where the sample was living, live cell culture or live pathogen, and 72% was using fixed samples, which speaks to the need for biological safety. Is it really important? Is it not important? In the sense that once the sample's already been processed and needs to be analyzed. But this is quite a big one. We always encourage in the project proposals to include preliminary data or something to indicate either the challenge or what's going on to understand the biology, and to include microscopy data is the cherry on top. Of the 63 that had preliminary data, 20% of them had qualitative microscopy images, where it was just an image with an arrow or something to indicate it had been done, or some phenotype was visible. There was zero quantification. There was no image analysis done. There was no kind of anything taken further beyond that simple image. So there's no necessarily understanding of what that image really portrays, other than a once-off random event that the person could have captured. Okay, so this was quite notable to us. And I think largely the most notable is that 80% of the applications had no preliminary data. So going from trying to do no microscopy or having never done microscopy to doing an advanced application is always going to be incredibly difficult, no matter where you come from the world. But in a resource-limited environment, that's even harder. So that highlights one of the key things that as a community, we need to find out how we can address. How can we make it easier to get preliminary data so that we can start developing the ecosystem that allows us to do microscopy research? Most of the applications sought to do confocal or wide field microscopy almost exclusively. For all intents and purposes, there was no application that had a good justification for a super resolution microscope, which might make sense, but we don't understand why. Is it because the research doesn't need super resolution, or is it because the researchers don't understand what we can do with super resolution? I think that's an interesting thing to try and understand. And in terms of the types of samples, 41% of the projects were related to tissues of some kind, either biopsies or humans samples taken from the clinic, or animal model samples, and then followed by bacterial culture. And then I think which stands in stark contrast to the research in Europe and North America, for example, is that cell culture, mammalian cell culture, was only 10%. Whereas I think often in the global north, that's the inverse, where most of the research is done in model cells before understanding human or the animal pathologies. And then this is an interesting graph that I haven't actually spoken about in any setting. And I wanted to discuss it here because this speaks to the type of thing that AMI is able to support, but it doesn't necessarily mean it's directly addressing what you do in your research. So the graph on the right indicates the total number of projects that fell into a category, whether it was screening or understanding mechanism, or if it was assessing drug efficacy or something along those lines, whether it was accepted or rejected. And the one thing that you will note is there was a huge number of researchers looking to do a characterization or an identification of some kind, okay? Now, think of this in the context of one lab on the entire continent where researchers are trying to come and look at a sample to just identify if something is what they think it is, if it contains what they think it contains, or what it looks like. That's nonsensical to have to travel all the way to Cape Town just to screen your sample, to look for something. It doesn't need to be done on an advanced microscope. It can be done on a good research grade microscope. So even though AMI exists, there's a huge gap in the pipeline, in the sense that not all of this stuff needs an African core facility, as was mentioned by you. A simple microscope put into the right place would be able to be much more impactful, in this case, in the entire AMI lab. And that's an interesting thought. So to back up my claim, this is the totality of the preliminary microscopy data that was included in all 63 projects. Okay? Pardon? 5 minutes. Okay. Did you say 50? Okay. All right, I'm going to speed it up a bit here. So I want to reiterate that getting preliminary data is incredibly challenging, and AMI is not going to be in a position where the imaging center can be the source of preliminary data. It doesn't make sense to fly somebody to Cape Town to get an understanding of what a sample looks like. But microscopy understanding, as I've mentioned, we don't know what is the limitation that no one wanted to do super resolution, is because no one understands or because there is no need for it. We, as AMI, we don't know, and we'd like to find out. Okay. I'm going to speed through a bunch of these, and I really do apologize. I hate it when speakers do this, so I really do apologize. So we're trying to bridge this gap. AMI exists, but we know it's not the full pipeline. So we need to get the microscopes where they're going to be helpful on the continent. And we've done this through two main mechanisms at the moment. One is Open Scopes Africa, where if you have access to a little bit of funding, you can build your own microscope. And you can reduce that cost because you can build the exact microscope you need for your application. And you can make that microscope cost 90% less than one that you'll buy from a commercial provider. Okay, so it's not a silver bullet that's going to solve all problems, but it's useful to know as a community that there are alternatives. You don't have to go to a commercial supply and buy an expensive microscope. You can build a microscope. It does take expertise, and that's what we're trying to support. There's also mechanisms through which you can take your existing microscope that might not have a camera, and if you spend $200 buying stuff from Amazon or what have you, you can turn that normal microscope that you use with your eyes into include a camera that you can then capture. And what you do with the captured image, you can share it, you can include it in publications, you can also analyze it. I don't even want to say on a basic level. You can do very good analysis on things. And it's about pushing the boundary of what we can achieve with the resources that we have. We have the program for equipment exchange and reutilization. So I will skip to this slide. We've helped facilitate the donation of 4 microscopes. There's a gap over there on the continent. I understand that even this process was a bit of a challenge because I think some of the microscopes were delivered with the incorrect components, but we're working on this. We're trying to get the microscopes to where these intermediate steps can happen and to a point where AMI can be more impactful as a continent-wide partner. All right, we have the online videos that I mentioned. So if you would like to brush up on a little bit of microscopy knowledge, we currently have talks on fluorescence, resolution, and confocal microscopy. These are one of our own, help contribute to the central one. And then the Partners in Teaching and Fellowship Program. This again speaks to the education, which is one of the things I wanted to highlight. This is an ongoing program that the applications closed, I think, a month or 2 ago. So unfortunately, if you weren't in time, potentially in the next run. But what we're trying to do is we're trying to pair African researchers with global microscopy experts. Get them talking, get them working over the course of 12 months to develop and understand what is going on in Africa and host workshops in Africa that can help training and can help educate. Okay. So again, with AMI, our vision is always multi-pronged. We're trying to get the equipment to where it's needed. We're trying to get the knowledge and the skills to where it's needed. And then in closing, my final thoughts, training and understanding is huge. But what is training and understanding? There's the training on what buttons to press to use a microscope. There's the understanding of how that microscope works and what I can do for it. There's also the understanding in the biology, which is a massive thing that not necessarily there is going to be an expert, and you might have to be that expert in understanding how your sample needs to be processed and how you can use a microscope to study it, okay? It's a bit of a spoiler for things that are yet to be spoken about, but the AIC and AMI and I4A, it's not enough, right? They're fantastic, don't get me wrong, and you must take those opportunities, but it's not enough to address every single researcher's need on the continent that needs a microscope. So we need scalability for exploratory or early efforts into microscopy, and we need to find better ways of doing this. Low-cost options, open scopes as an example. What about regional hubs where knowledge and expertise can be shared more efficiently? These don't have to be new facilities. They can be existing facilities that can help facilitate their immediate location and helping disseminate knowledge. Funding is key, okay? One of the reasons why tomorrow we'll be writing a writing workshop. It's up to us. There's no one giving us handouts, right? We as researchers on the continent need to fight for what we need to do. So we have to be strong and competitive in seeking out our funding. So it starts with understanding and having clarity in what you want to do and what you want to achieve and how you want to do it. So again, this reinforces the need for understanding and education. If you understand the research that you want to do, you can better sell it, right? And this, once again, justifies and reinforces the need for more training on the continent. But most importantly, despite everything that's going on in the world, together we are stronger as an African community. The more we engage as ABIC, the more we engage as local communities. I know Kenyan Bioimaging, for example, has started up recently. We benefit from one another and we are stronger together. And I think we must do whatever we can to maintain these networks that we establish through ABIC and find ways to make them stronger and reinforce them. So I hope that was not too fast. Apologize for going very quickly. Not to draw it out at all. I would really like to thank everyone involved at the AUC, everyone that's been involved in ABIC coming to Cairo. Thank you so much. To everyone. I'm not going to list everyone because I'm going to speak too long, but thank you on behalf of me. Thank you very much. And I'll be happy to address any questions.